Good morning, children. Yesterday we did cytoplasm. So today we will continue with the same. It was little left. So we are going to continue with the cytoplasm. Yesterday we were doing the functions of cytoplasm. And in the functions of cytoplasm, we have done the cytoplasm. The first function we did that it is a site for organelles and inclusions. That organelles are present there, inclusions, cell organelles and cell inclusions are present there. It has enzymes for various metabolic reactions. So in functions we can write down. I'll write the functions again for you so that it comes on the single page for you all. So here the first function, it is a site for cell organelles and cell inclusions. You all know what are cell organelles and cell inclusions. Yesterday I told you what are cell organelles and what are cell inclusions. Yesterday I told you this. So I think you know organelle first. Tadarani, shall I unmute you so that you can speak? Just a minute. Let me set the camera once more. Yes. Yes, Tadarani. You can write only. Yes, you can text it. Yes. Example of cell organelle. First, whosoever knows example of cell organelle and then example of cell inclusion. One example each. Yes, who's going to answer this question? Yes, beta, I'm waiting for the reply. Example of cell organelle and example of cell inclusion. Mitochondria is the example of cell organelle. Yes. And Rohan is saying Golgi operators. Okay. Yes. Cell inclusion is lipid. You cannot say globule, glycogen granule. Yes. Yashika is saying lipid glycogen. Yashika, glycogen granule or lipid globule. These are cell inclusion which has these biomolecules in them. Like it is a body, it is a small structure in which lipid is present. We call it lipid globule. If it is a small structure in which glycogen is present, we call it glycogen granule. Lipid molecules, lipid globules is a better word. Okay, so these are cell inclusion. Now this is the site where cytoplasm is the functions of cytoplasm. I'll write here functions of cytoplasm. So functions of cytoplasm, it is a site where cell organelles and cell inclusions are present. And the second is, it is a site where many enzymes are present. Enzymes are present here for various metabolic reactions. Are present here. Here. That means the enzymes for various metabolic reactions are present here. For example, and translation. Translation is the process of protein synthesis. Translation, this you will study when you will reach class 12. Translation process in detail, a big chapter on this. So translation is the process by which protein is synthesized. But glycolysis, you already know, glycolysis is a process. What happens in which pyruvic acid is formed, okay, by the breakdown of glucose. It's a 10-step process which we have already done in photosynthesis in higher plants, okay. So this is the site where enzymes are present. Then, it helps in proper distribution of substances. The substances are properly distributed in the cytoplasm. So we can write down the third point, helps in proper distribution of substances. All the substances are properly distributed here. The fourth point is it shows the process of cyclosis. Shows the process of cyclosis. Cyclosis, as I told you, it is the streaming movement of cytoplasm. So yesterday also I told you streaming movement of cytoplasm. Streaming movement of cytoplasm is cyclosis. So it shows the process of cyclosis. This process of cyclosis is of two types. This is of two types. The first is rotational, another is circulation, circular. So rotational, the first process is rotational. And the second type of cyclosis is circular. What happens in the rotational cyclosis and what happens in the circular cyclosis? Suppose this is a cell. I'll make it here. This is a plant cell. I'm making hexagonal. And it has, I'm not making cell wall or cell membrane because our concern here is cytoplasm. So here we have a big vacuole in the cell. You know the plant cells, they have big vacuoles, okay, which push the nucleus on one side. So the cytoplasm in this moves in this direction. In one direction, it keeps on moving. And this movement is known as rotational cyclosis. And in animal cells, what happens? Animal cells have many vacuoles. One vacuole, another vacuole, another vacuole. So there are many small sized vacuoles in animal cell. And the cytoplasm in animal cell, it moves around each vacuole. Suppose it is moving like this around this vacuole. Okay, it is moving like this around this vacuole. Cytoplasm is moving like this around this vacuole. Here it is moving like this. So this is the movement of cytoplasm in different direction around very small vacuoles. So movement is, you can write here movement in one direction. Movement, I'll write here so that I get sufficient space to write on the next point. So movement in one direction around a large vacuole is rotational. Movement in different direction around small vacuoles. Movement in different directions around many small vacuoles. This is circular cyclosis and rotational cyclosis. So we have learned two types of cyclosis here, circular and rotational. We come to the next one. helps in pseudopodia formation. Now, anybody who knows what is pseudopodia? Anybody who knows what is pseudopodia? Yes, who will answer this question? What is pseudopodia? Yes, Peta. False feet. It is formed in which case? Yes, it's a protrusion of the wall, the cell, the plasma membrane. In which case, amoeba. Yes, yes, Priya Yadav helps in capturing food. Amoeba. Yes, yes. So helps in formation of pseudopodia. Pseudopodia is seen in amoeba. So amoeba is a unicellular animal, so it's a cell. We have nucleus, okay, and we have cytoplasm, in two forms. In two forms. Outer here, the outer which is present towards the periphery. This cytoplasm is thick and viscous. This is thick and viscous. That is why I'm drawing it, little more of the spots I'm drawing to show that it is thick and viscous. Whereas the cytoplasm which is present towards the nucleus, it is thin and less viscous. So this is known as the outer, which is thick and viscous, is known as ectoplasm. Ectoplasm, which is thick and viscous. Whereas the inner one, which is not dense, this is known as endoplasm, which is less dense and less viscous. They have been given another name also. The outer one, which is thick and viscous, is also known as gel, G-E-L. And the inner one, which is not dense, don't write less, it is not dense at all. Okay, not dense is known as soul. So we have two types of cytoplasm. One is towards the periphery, and another is towards the nucleus side. One is dense and one is not. One is the one which is dense is known as ectoplasm or gel. The one which is towards the interior side is known as soul, and it is not dense, and it is thin, and it is not viscous. Okay. Now, what happens that these two forms of cytoplasm are interconvertible. That means gel can get converted into soul, and soul can get converted into gel. So they are reversible. Gel can get converted into soul, and soul can get converted into gel. That means if one is thick, viscous, dense and viscous, it can become thin and not dense, and the one which is not dense and which is not viscous can become viscous and dense. So these are interconvertible. Now what happens? Suppose, now I'm drawing here only. Suppose this is an amoeba and it has to make a false foot. So what happens? That suppose it has to make a false foot here. The thick and viscous soul here, it gets converted into the thick and viscous gel here gets converted into soul, SOL. That means less viscous. It becomes less viscous. Now, when it becomes less viscous, the cytoplasm moves out. The cytoplasm moves out and sends out a protrusion like this. Okay. Suppose here also one protrusion needs to be formed. So here also the gel will become soul and it will make a protrusion. And later on, what happens? What happens then? Then this will move towards each other like this. It will move towards each other to capture that food particle, and a complete vesicle is formed, and it is then taken in. So this is how the formation, the conversion of soul into gel and gel into soul helps in the formation of pseudopodia. Okay, the something which is soul-like, it soul. When it is soul, it flows freely. When it is gel, it is not able to flow freely. So that is why when pseudopodia has to be formed, at that time what happens? The gel gets converted into soul. It flows freely and forms a protrusion, which is a false food. Is it clear, children? This functions of cytoplasm clear? Functions of cytoplasm is clear? Yes, I'm getting one response. Yes. Functions of cytoplasm clear, beta? Okay, fine. I come to the next topic now, and that next topic is endoplasmic reticulum. Now we move on to the cell organelles. Now we will move on to the cell organelles, and the cell organelle, the first topic, the first cell organelle which we are going to study is endoplasmic reticulum. The one which we are going to study is endoplasmic reticulum. Yes. So let's start with the first organelle. Endoplasmic reticulum. This endoplasmic reticulum, have you heard of this before? Have you heard of endoplasmic reticulum, beta? This is a cell organelle. Is it membranous, membrane bound, or non-membrane bound? Okay. Is it membrane bound or non-membrane bound? Who's going to tell me? Non-membranous. Amrita is saying non-membranous. But it has membrane. It has membrane. Cell organelles, all the cell organelles except for ribosome, which we do not consider in cell organelle, they do not have to have membrane. Otherwise, all the cell organelles have membrane. Some of them, they have double membrane. Can you name the organelles which are double membranous, which have two membranes? Can you name the organelles which double mitochondria? Yes, mitochondria is double membranous. Chlorophyll is also double membranous. They have their own this DNA also. Circular DNA is present in mitochondria as well as chloroplast. Yes. So these are double membranous. Whether it is endoplasmic reticulum or it is lysosomes or it is Golgi complex, they all are membranous organelles. They all have membranes around them. Okay. So this endoplasmic reticulum was first of all observed by Garnier. This was first of all observed by Garnier. And the ultrastructure was given by Porter. He was the one who gave the ultrastructure of the endoplasmic reticulum. This endoplasmic reticulum is absent. The prokaryotes, they do not have any membrane bound organelles. You know it very well. Prokaryote means, if you take example of bacteria, they do not have any membrane bound organelles. All the prokaryotes, this is a feature, characteristic feature of prokaryotes, that the nucleus is indefinite without nuclear membrane, and membrane bound organelles are absent in prokaryotic cells. So absent in prokaryotes. Absent in prokaryotes. Present in eukaryotes. This is a membrane bound organelle. I'll write here so that you understand it. Membrane bound organelle. Present in eukaryotes. Now, is it present in all the eukaryotes? Is it present in all the eukaryotic cells? No, except for mature red blood cells. The mature red blood cells, they do not have any organelles. So this organelle also is absent in mature RBCs. Otherwise, it is present in all the eukaryotes, all the eukaryotic cells. Now, you know, when we talk of muscles, in muscles, ER is known by the name sarcoplasmic reticulum. In muscles, this is known by the name sarcoplasmic reticulum. And here in muscles, it helps in contraction. Okay. Now, it helps in contraction of the muscle. And it is in muscles, it is also known as, in muscles, it is known as sarcoplasmic reticulum. Now, components of ER. What is ER made up of? Components of ER. Endoplasmic reticulum is made up of 3 components. The first one is cisternae. The second one is vesicle. And the third one is tubules. This is made up of cisternae, vesicles, and tubules. Now, what are cisternae? What are vesicles? And what are tubules? I'll tell you. Okay. See. Here, we will take up the first one, which is cistern. These are unbranched, lie near the nuclear membrane. Lie near the nuclear membrane. Why do they lie near the nuclear membrane? They lie near the nuclear membrane because they are formed from the nuclear membrane. If this is a cell, this is the nucleus of the cell. You know, the nuclear membrane is double walled. You all know this. This is the nuclear membrane. Nuclear pore is also there. If this is the nuclear membrane, it is formed by protrusion of the nuclear membrane. That means this nuclear membrane protrudes out. Protrudes forms the Yes, that is why we say that cisternae are present near the nuclear membrane, because they are formed by the protrusion of nuclear membrane. They are unbranched and they have these bead-like structures on them. You know what are these bead-like structures which are present on them? Yes, can you tell me what are these bead-like structures which are present on them? I have made some bead like some round. Yes, Sanvi Rana, you are correct. Rohan, you are correct. These are ribosomes. So here the ribosomes are present. These are ribosomes. These are ribosomes. And these ribosomes are attached to the membrane of cisternae by a protein. And the name of that protein is riboforin. So ribosomes are attached to the membrane of endoplasmic reticulum by the help of a protein called riboforin, PHORIN. So this protein riboforin helps in attachment of ribosomes to the membrane of endoplasmic reticulum. So we have this. This is about the cisternae, which forms a important uh, component of endoplasmic reticulum. One more thing is their lumen present on it. This cistern is the inner, this cavity is known as lumen. This is known as lumen. This lumen of cisternae, lumen is small. Lumen is small. The lumen of cisternae is small. So this is another feature of cisternae. Okay. Then we come to next, which is vesicle. So number 2, we will start with the next one, which is vesicle. Vesicles are small, round shaped oval bodies. Okay. Small, round or oval structures found near cisternae or tubules. They may or may not have ribosomes. They may or may not have ribosomes. So where are these vesicles present? So see, vesicles are present here, vesicles are present here, vesicles are present here, vesicles are present here. Some of them can have these ribosomes on them. Some of them can have ribosomes on them, like this, and some may not have. So these are small round oval structures which are present near the cisternae or tubules. They may or may not have ribosomes. Now, the third structure which is there is known as tubules. So we come to the third structure, tubules. Vesicles, cisternae, and this one is the third one, which is known as tubules. Now, what happens in case of tubules? Tubules are branched. They are branched. Unlike the cisternae lies near the nucleus. Now, cisternae lie near the nucleus. You must have seen I have drawn here cisternae which lie near the nucleus. These tubules, they lie near the plasma membrane. Okay, they are located near the plasma membrane. They are located near the plasma membrane. They are branched. They are located near the plasma membrane, or they are located close to the plasma membrane. And in the tubules, no ribosomes are present. No ribosomes are present. Ribosomes are not present. At the same time, one more point is their lumen is broad. They have broad lumen. Broad lumen. That means they look like this. And that means if this is plasma membrane, they come out of it. Okay. They do not have any of, and then from here they are attached to these cistern. They are attached to cistern. These cisternae have the ribosomes on them. They do not have. They have ribosomes on them. And where are they attached to? They are attached to the nuclear membrane. This is the complete cell. So they also give rise to vesicles. They also form vesicles. Their vesicles are without the ribosomes, and the vesicles which are formed by the tubules, they have ribosomes on them. So these are tubules. I'm writing here. These are tubules from here to here. These are vesicles. The round ones are vesicles. These are vesicles. And these are cisterns. Bache, is it clear? Bache, is it clear to you? Is it clear, beta? Is it clear? Okay, I'm getting the response that it is clear. Okay, so these are the components, and as I have told that the components are cisternae, vesicle, and tubules. Vesicle are formed, are sac-like structures, small, round, oval sac-like structures, which can be formed from the tubules as well as from the cisternae. Some of them, they have ribosomes, some of them do not have ribosomes. The cisternae they lie close to the nucleus, and the tubules, they lie close to the plasma membrane. The cisternae, they have ribosomes which are attached on their surface by a protein named as ribo. What is the name of the protein? Ribofourin. Ribofourin. Their lumen is small, and the lumen, tubules, they do not have ribosomes on their body, on their surface. They lie near the plasma membrane, and their lumen is broad. Their lumen is broad. So these are the three parts, three components of endoplasmic reticulum. Any problem till now, whatever I have taught in endoplasmic reticulum or in the cytoplasm which I taught? Yes, any problem in that, beta? In both the topics, any query? Yes, bache? No doubt. I'm getting the response. Rohan is saying, no doubt, ma'am. Yes. Sadarani, no. Okay. Sanvi, Harsha, Yashika, Priya, Yashika, clear. Shiksha, no doubt. Okay, Shiksha. Others? Lalit, boys do not respond. Lalit, Sarthak